Aloha everyone, and thank you all for joining us for our weekly Hanauma Talk series. Tonight I'll be introducing Shimi Rii. Is that how you say your last name? Yeah. And she'll be talking about the Heʻeia National Estuarine Research Reserve Program. By the way, my name is Gavin Y. I am the Outreach Coordinator for the Hanauma Bay Education Program. We'll have a three consecutive talks, one after another, and Shimi will introduce our next two speakers after her talk. So without further ado, Shimi. And for those of you who are joining in, can you guys turn off your mics and your video function? We appreciate it. Go, Shimi.
Okay, thanks, Gavin.
Yep.
Can everyone see the screen? And then I want to have this open. Okay, so aloha everyone. Thank you for being here tonight. My name is Shimi Rii, for those of you who don't know me, and I'm the research coordinator for the Heʻeia National Estuarine Research Reserve, and I'm also based at the Hawaii Institute of Marine Biology. And today I'll be giving an overview of the research and monitoring program of the Heʻeia NERR, or the National Estuarine Research Reserve, and provide a description of how we strive to conduct research in Heʻeia, and I hope I can set up some background for Aka and Sarah. Last week, if you guys tuned in, Fred, who is our education coordinator, gave a wonderful presentation about the past and present of Heʻeia. So I'll be building off of that and describing how we're tracking changes in the Heʻeia Ahupua'a. But before I move on, I want to point out that this picture was taken here. This one was taken by Keli'i Kotuvete at Paipai'o Heʻeia. This is actually us off of the Kahokele Makaha at Heʻeia Fish Pond. It's actually myself, Aka, who's going to be speaking next, and then two of our technicians. And we were building one of our long-term water quality monitoring stations to monitor the water characteristics that's going in and out of the fish pond. So that's one of the things I'll be talking about today. Okay, so I think with this crowd, I don't really need to talk much about the Ahupua'a, but just to get everybody on the same page, it's basically a sustainable management system using natural watershed boundaries that Native Hawaiians used from Mauka to Maikaʻi. It's a resource management system that basically they understood that there were lots of nutrients and cold water that was coming down from the mountains, and they used flooded field agroecosystems to capture sediment and grow some food. And then they were also able to use coastal fish ponds or some local vie systems as a nutrient sink, also to maximize primary productivity and provide food for herbivorous fish. And then this way they were able to keep the shore ecosystems healthy and food web dynamics healthy for ocean life. So in this vision of the Ahupua'a, it's really aina momona. For those of you who've heard of this phrase before, aina, that translates to that which nourishes and the abundance of the land, the abundant legacy. But when we think about aina momona, it's really looking towards the past, when Native Hawaiians were able to sustain themselves and their population for centuries and for a long time. But currently in Hawaii, Oahu has lost over 65% of its wetlands. Hawaii now imports over 90% of its food in an island ecosystem. So what is really our vision of aina momona now? And how can we look towards creating aina momona in the future? And I want to take just a few minutes, maybe just a minute, to ask you guys on this talk today, what do you think of when you hear the phrase aina momona? And if you guys can put that into the chat, just a few phrases, or if it's like species abundance or biodiversity, or if it's just kalo, if you guys can chat some things. Protecting the land, thank you, Kuamo'o. That is a really good one. Anyone else? Bountiful, yep. Maya says abundant. Any of your favorite species that you think of? Richness, yep. sustainable is a really, really good point, right? You can't have aina momona continuously without sustainability. Part of who we are as a people, abundant and good people, aina relationships. So that's a really good point, Lehiva, and I knew I could count on you, because that's my next point, is that when you think about aina momona, you think about the abundant fish, the abundant kalo, abundant food, but humans are this key partnership in maintaining the aina momona, right? So basically Native Hawaiians had a culture that was rooted in reciprocity. You gave to the land took care of you. And for the Heʻeia NERR, we really want to think about how can we conduct this research or science and track science in this environment through reciprocity. How can we give back to the land while the land gives knowledge to us? So yeah, if you keep coming up with good things, feel free to put them in the chat, and we can all learn from each other. But how do we accomplish reciprocal collaboration in science? This is one of the things that we thought a lot about. We framed it in this paper, but we're also greatly guided by the Kulana no I'i, which was a collaboration between Sea Grant College and Kua'aina Ulu'auamo and Pai Pai and many people that was involved in stakeholder meetings and talking about how to create these standards for building community researcher partnerships. But it's all about building relationships and taking the time to get to know the land that you're studying. So this community researcher partnership driven action, this is what Fred showed last week, but this is what drove the people of Heʻeia to nominate Heʻeia as a National Estuarine Research Reserve. And basically, what is the NERR? People always say the NERR, we're part of the reserve. The NERR is actually a 29 reserve, we're the 29th one, consortium. It represents 23 states and including Puerto Rico, protecting more than 525,000 hectares of estuarine and coastal habitat. And it was established in 1972 through the Coastal Zone Management Act of 1972. And South Slough in Oregon was actually the first reserve, which I didn't know. I always thought it was like one of the New England states. But this is how we came to be the 29th NERR. Our state partner is HIMB, University of Hawaii at Manoa, which oversees the partnership with NOAA. And then we have other partners that are listed here below. And then there are research, education, stewardship, and training. That's basically skills building and knowledge exchange. And we all work very closely with each other to accomplish our goals. So just to orient you, we're located in the Ko'olaupoko Moku, and there's in the Heʻeia Ahupua'a, there's two streams, Ioleka'a and Haiku, that merge into Heʻeia wetland, and then Heʻeia fish pond, and then into Kaneohe Bay. And then the Heʻeia NERR site is actually located kind of in the lower regions of the Ahupua'a. And there, as you can see, there's multiple different landowners and managers that are involved in the NERR site. And Fred did a great job talking about how mangroves are introduced in the 1920s, and misuse and disuse and abandonment of the area, caused some of the grasses, the invasive grasses to grow. And basically, since the early 2000s, many of the community groups and indigenous resource management groups have done a lot of restoration. And there's all these volunteer groups that come out, and it's because of that they've come this far into rebuilding the walls and rebuilding the agro ecosystem that's in Heʻeia right now. So basically, we want to restore ecosystem services to enhance resilience through indigenous resource management. What do you mean by ecosystem services? I'll kind of describe that briefly in a little bit, but when we think about it, we want to restore this. He showed this picture in 1928 in Heʻeia. This was the agro ecosystem right here, and then there's the fish pond right here, and then there's all these ecosystem services that we can that was tracked and was provided in this picture. But now when we restore, we have to kind of think about these stressors and changes in climate, such as enhanced storms and sedimentation and macroalgal growth and all these things that we have to keep in mind as we restore the ecosystem services. So just really briefly, ecosystem services as they're defined are kind of broken up into these provisioning, which is like, kind of like food or what supports you, supporting and regulating, which are kind of like habitat health and sediment retention and storm. And then there's cultural well-being that's all represented in ecosystem services. But this definition is very one way. It says conditions and processes through which natural ecosystems sustain and fulfill human life. But from what we're talking about with reciprocity, we want them to engage in these reciprocal relationships with humans. So we're trying to follow this Hawaii-based framework of cultural ecosystem services that has all these different categories that talk about exchange and connections between the land and the ecosystem. So when you look at something like this, where there's a big storm, and we have the fish pond becomes kind of a sediment retention area. So we think, okay, this is how we can study the area, maybe, right, with this supporting habitat health, sediment and nutrient retention, diversified crop production, well-being. But when you overlay the Hawaii-based framework, it gets more complicated. We have the spiritual landscapes, we have social interactions. It just gets really complicated, and it's not like, we can put them into 4 categories that we can study. So we wanted to make sure that our stakeholders' voices were heard. So we had a lot of interviews with people and stakeholder engagement and meetings and workshops, and we came up with these priority research needs that are kind of broken up into these branches right now. But of course, these branches are forever growing, and we're trying to find out ways to connect research through all of this. And what's really cool is that we're really, these questions for each of these branches are co-developed with our partners and managed and through all these meetings. So all of this came from collaborations within sector, within our training. We work a lot with Katie Hinson, who is our coastal training program coordinator, also at Sea Grant. And then Fred and I work really closely together to maintain collaborations. So each of these students contribute to the research that is, displayed in these branches. Last week, you heard from Evan, who is studying carbon and oxygen dynamics. Today, you're going to hear more from Aka and Sarah, who has their own, research that's very important in these branches. And then next week, you'll hear from 3 of our students and fellows, Matt, Melissa, and Alicia, about some of their work in the wetlands. So that's really, really cool. And then just really briefly, these are some of our monitoring sites. And this is not, of course, everything that we alone do. This is a big collaboration. Some of these sites are historical. They've been taken, throughout the years, since early 2000s. And so there's time series in the fish pond that the Allegado Lab continues. There's reef monitoring sites, there's wetland sites, there's vegetation sites, there's water quality sites. And so there's, as you can see, there's a lot of tracking, and it is also a task to connect all of the results together and try to analyze it. Just a little background on here. There's this is kind of our water quality sites. You can see that we're trying to follow the stream Mauka from the fish pond area all the way to Papahana Kuaola. And these are some of the water quality monitoring sites that we put together. That's installation. So like in Kaho'okele, this is something that we have installed out there. And this is a YSI multi-parameter SON measuring all of these things. So if you look at something like this, you can see temperature. This is just briefly, I don't want to take up too much time, but you can see that in the wet season, temperature is warmer in the ocean, but during the dry season, it starts to heat up on land. So you can see temperature increasing. And then with turbidity, which is kind of like how turbid the water is, we can see that turbidity increase is kind of heavy in the wetland area, but then during the dry season, there's more turbidity closer to the shore and in the wetland area. And this is just to show some of the inorganic nutrients, and I don't want to get too deep into it. I don't want you to look at some graphs, but I just want you to show that basically there's these big nutrients that's being taken up that are produced in the upper wetland that goes down. And these are the kind of nutrients that's really important for people like Sarah and Aka's study, because they're studying how these nutrients are consumed and are used up by organisms in the wetland. So we're trying to look at short and long-term change. And I think I'm around 15 minutes. So I'm gonna just skip through some of these, but I just wanna show that we look at vegetation as well after mangrove removal, and that's really important because we, now that the vegetation is changing a lot because of all the mangroves being removed, we're looking at water birds and how they're hanging out in the mangrove. And this is something that next week, some of our students will be talking about. We see these IO that are Hawaiian stilt that are using the lo'i, and basically she found out that the IO utilizes mostly all stages of lo'i cultivation for foraging and nesting. And then finally, we do some fish monitoring. We're doing all sorts of fish monitoring, and it's because we want to look at auaneʻi, and this is what Aka's going to talk about next. He's going to talk about some of the prey items for auaneʻi, which is our native striped mullet. And there are, and then we're also looking for, in fish monitoring, we're looking for o'opu. Many of you who might know o'opu, they're these really charismatic goby species that have these suckers and they climb up waterfalls, some of them, and there you can see the distribution of native o'opu. And in our wetland, we were able to find actually o'opu nakea in the upper watershed, akupa in the lower watershed, and naniha in the lower watershed. So that was really cool, and we're hoping to see more of these species come back. So finally, aloha aina, when the pandemic is over, please come out and help us out in Heʻeia and lend a hand. And these are really the pictures of, images of people helping out and making this place the beautiful place that it is. So I just want to end with, I hope you can take home thinking about the phrase aina momona and going back and asking your families and friends about the role you can play in perpetuating and sustaining aina momona. Okay, so this is a o'opunakea from Papahana, very, very cute. And next we're gonna have Aka Beebe, who is a master's student in UH Oceanography, and he's going to talk about some of the work he's been doing at the fish pond.
All right. Hello, guys. Hello, my kako. Can you guys hear me? Can everybody hear me? All good?
Aka,
All
we
right,
can hear you, but it's your slides.
Oh, gotta present them. Mahalo for that.
Yeah,
mahalo for that great introduction too, and getting us all geared up to think in the aloha aina momona sense. Eyo, and I'd like to start. My name is Aka Beebe. Currently, I'm a master's student in oceanography, being funded by a fellowship through the Heʻeia Estuary National Research Reserve. That's a mouthful. So let's begin. E hinu auaneʻi na nuku he maikaʻi ko laila. So I wanted to start off with olelo noʻeau, and it's one of the one of the wise sayings that's historical. It's cultural. It's Hawaiian, and what it's saying here to me, and I hope so it gleans a little bit more truth to you guys, especially being in the aina momona aloha aina mindset, is that where the mouths are gleaning with fat, then you know they're eating good. So right here is a picture of aholehole. Not really the target species I'm talking about, but I like this picture. They have beautiful eyes and nice fat lips. So this is the nuku hinu, and right here, this is something that caught my eye. This is the maikaʻi. So what we see here is something that caught my eye while walking around the Heʻeia fish pond on the kuopa at one of the makaha. It's some pua'ama, or I guess young mullets, fingerlings. And what they're doing, they're actively swimming up and down through the still water, You know, it wasn't a very hot day. There's flow through the makaha, so I'm assuming there's a lot of oxygen, but I hypothesize that they must be, you know, doing this for a reason, and I think they're eating, you know. It's the po maikaʻi. They're eating the things that we don't see, and these are the important guys when you're talking about fish pond production. In the loko i'a, they target mainly lower trophic level species, so species that graze on species that eat phytoplankton and other primary producers, macroalgae. They also can feed on other smaller items, zooplankton, that can support, you know, the larger fish, larger metazoans, and ultimately they can support us. So I'm going to talk about what these guys might be eating in a little bit. So thinking about loko i'a and what they were designed for, they were designed for supplying food. You know, that was the necessary function that needed to be done back in that time when there was no mats and containers and stuff like that. So they're an invention of necessity. They're a unique creation in the Hawaiian Islands, and a lot of them have been left derelict and damaged, but there's a revival of them being restored and being put back to use. And at one of these sites at Paipai, or at Heʻeia Fish Pond within the Heʻeia watershed, they hold the spot of restoring a working loko i'a. Paipai o Heʻeia, they're the kiaʻi, the caretakers, the stewards. So yeah, so loko i'a, providing fish. Most of the time, it's not a lot of big time input of food or nutrients from like fertilizers and stuff like that, which you may think about when you think about aquaculture, like industrialized aquaculture. Mainly, they're engineered to look at the resources that are available and see how we can amplify those and how we can produce that momona, you know, how we can keep it all together and still have a resilient system. So one of their main goals is to produce these primary producers, phytoplankton, macroalgae, and one of the guys I'm going to be talking about, or I'm focusing about, is the diatoms. So just a little background, people from all over here. So primary producers, they're the base of the food web. They're bringing the food into the system. So the system being all us guys that need to eat and we cannot make food, you know, like, so they're making food out of the sunlight and like rock and water. And so they're supporting the food sources of our food. So if you can imagine that previous slide with the pua'ama, they're eating stuff that we can't see, the primary producers, and they hopefully will ultimately end up on the dinner table and in our stomachs. So yeah, with that, loko i'a have the potential to provide food and serve another necessary purpose in this modern day of feeding us all. Moving on. Okay, so the two players that's in this story are the ama. The striped mullet is a native species of Hawaii, and it's also found all over the world. But this guy, you know, this guy got here just like any other native species. One of the target species in fish pond productions, as I mentioned, and they primarily feed on diatoms found on benthic surfaces, so at the bottom of the water column, where you can find sand, sediment, you know, mud. They can sift through that, and also they can graze in the water column. And diatoms, which are a main component of the mullet's diet, they're phytoplankton, and they're pretty important. They're found all over the world in all types of environments, not only in the ocean. Pretty much if you have any trace water, you'll find diatoms, like even in ice sheets or on rock walls. They're estimated to produce about half of the oxygen from the ocean. So that's kind of a big deal. They also have a unique characteristic of this outer covering called the frustule that's made out of silica. That's another important thing to think of because silica is the rock. And then they have rapid response to environmental changes. Although we can't see these guys a lot of the times with our naked eye, we can see them when they're, you know, just cranking, they're going off, they're blooming. Whenever the environmental measurements, the environment changes, they'll bloom when it's in their favor. So keeping those guys in mind, my research, I would like to find out how diatom abundance and distribution is affected by environmental conditions of the local ia, being Heʻeia fish pond. So that's a big part of my research. And also because we're dealing with a time component because of the time series, I'd like to also see if there's seasonal variability in diatom abundance. And I know it's you know, it's a question that a lot of people ask, and I know there's a lot of ways to answer it. And I'm just going to show one way that I tried, fast kind. And hopefully we can look at different ways of breaking up seasons and stuff like that in the future. Okay, so going into my sampling design, here is a map of the site. Okay, so here's Heʻeia fish pond. This is Heʻeia stream, connecting it to the Heʻeia watershed and the wetland, Kapoʻiwi, and the green sites are ones that are designated local sites. They're within the fish pond. The red sites are ones that are designated as makaha because they were actually taken right at the inside of the makaha. And the yellow sites are the two end members I selected, one being a representative of freshwater, straight from the stream, and the salty ocean, as salty as I could get on the sampling day, which is at the edge of the reef right outside Kahoʻolahui. There's also one more site that I sampled. It's a newly constructed pua pond. So it's pretty much a nursery for the pua ama, for the pua, the baby fish. So it's a pond that they, you know, they populate with hatchlings or fingerlings from the tanks, and they grow them up from there. I measured a lot of different parameters: salinity, water temperature, DO, conductivity, salinity, and pH. I also, from bottle samples, process them for nutrients and for genomic DNA. And okay, so going into some of the characteristics of Heʻeia fish pond, it pretty much resembles an estuary, and that makes sense because it's at that confluence of freshwater and the ocean, right at the, actually the stream comes out here. This is a bad map. Don't blame me, it's USGS. So the salinity at different tides changes, as does in estuaries. There's also some degree of stratification in some parts of the fish pond. It also depends a lot on wind action. So the wind can really stir it up. It's very shallow. It's like less than 1 meter in most parts. At low tide, some parts can be almost exposed. If it's real low, then yeah. But what's good about you know, the construction of this and the engineering is that there's always adequate flow through all the makaha at all tidal lakes. So that's a good thing. There's always that exchange of fresh water and ocean water. And looking at temperature, there's a seasonal signal in temperature. Kind of a small signal, but albeit it's different. And nutrient concentrations, this is aggregated across the entire fish pond. So not much difference, so little blips here and there. And definitely if we had greater resolution or more uniform sampling, it would have been a little bit more telling. So getting into what I'm doing, measuring those parameters and collecting bottle samples. From there, I extract the DNA, And from that extracted DNA, I use a technique called qPCR. And I'm using I'm using primers that are specific to just diatoms. So they're just going to be counting diatoms. That's going to be the main measurement that I'm getting out of it. And yeah, so I gotta count all these guys without even seeing it. So let's get into it. Okay, so looking at some trends, we have a plot of salinity and diatom abundance. The x-axis, I have different bins of salinity, so just different groupings of the salinity found throughout the fish pond at all the sample sites. So here we have the very fresh from 0 to 10, and at the other end we have very oceanic 30 to 40. And these plots, and on the y-axis here we have the diatom abundance, and the numbers are in cells per liter. So it goes from around 10,000 to 10 million. And that's a pretty big range. So some little differences are actually pretty big. So if we look at these plots, what is happening is the boundary between the boundaries of this blob, they call it a violin. It indicates how many sample points fell within that measurement range. So about little less than 1 million, most of the sampling points, that was the measurement at this salinity. So at the very fresh water, most of the measurements were less than 1 million. And if you go up in salinity, it kind of changes just a little bit, getting closer to 1 million. This one, it dips back down, but it stretches out more in the extremes to have higher abundances, higher counts. And this bin, it includes the reef. So that one actually, yeah, it contributes a lot more lower measurements because of that. And now looking at each site, here I have the sites on the x-axis and the same diatom abundance on the y. The sites are sequenced pretty much in salinity. So going from low salinity to high, the colors also represent that. So the darker blues being low salinity and the brighter blues being the high salinity. So at the two ends, we have the freshwater Heʻeia stream sample, and we have the edge of the reef oceanic end member. And you can see all these sites within the fish pond are generally the same. Some lower measurements at the more freshwater influenced sites. But right here we see the pool pond, which is kind of an outlier because its physical environment is really different and it is constructed for a different purpose. So this might be an indication of whatever is happening is working because there's a lot of diatoms in there. So now, because of the differences not being so pronounced in the last two slides, I tried to look at the seasonal variability and break it up that way. So I broke it up between dry season and wet season. And given the sampling dates that I had, I quoted the wet season as December through March, and the dry season includes April, June, and October. So the colors again represent salinity, and interestingly, during the dry season, salinity was a little lower, which kind of goes counter to what I was thinking. With more rainfall, it would contribute more stream flow and therefore lower salinity. But anyways, we can see that the range in diatom abundance is much greater during the wet season, and there's a lot more measurements in the higher range compared to the dry season. So this line right here is like this is 80% of all the data is up to here. So of all the measurements. Oh, sorry, this is 90%. So only 10% of the measurements are in this end. Okay, and now breaking up the seasons to look at salinity differences, we can see this interesting pattern where the mid-range salinities have higher abundance in diatom. And now looking at the sites, breaking it up between seasons, we can see a little increase in all the sites. Some of the sites decreased, but generally speaking, there's an increase, especially in the pool pond. Okay, now looking at spatial trends, here we have all the sampling sites, and I took the measurements of diatom abundance, and I pretty much connected the dots. I mean, I didn't myself, the computer did it. But here the colors represent diatom abundance. And on the scale, we can see, that the dark purples are from 10,000, and it goes all the way up to the reds, which are 10 million. So with all the dates included, you can see the distribution of diatoms across the fish pond. And now if we filter the different seasons, if we look at just the seasons, we see differences in the distribution pattern. So at this side of the fish pond, this is where the stream is coming in. So this is more freshwater influenced. It would be really cool if I could overlay something that would convey salinity to, but here, take my word for it, it's fresher. And also we see a lower abundance in the mid pond and towards the Maka end on the south side. During the wet season, there's higher abundance on this freshwater influence site region, and not many dips in abundance throughout the fish pond. So we can see that the patterns in distribution change throughout the seasons throughout the fish pond. So that's pretty interesting. Okay, so in summary, yeah, diatoms can be variable throughout the fish pond, and I've shown that there's some physical measurements and chemical measurements that can predict how they will change throughout the environment. So if you have an environment with low salinity, most likely you'll have less diatoms there. But it could just be because of Heʻeia fish pond. That's what this study is focused on. I'm just looking at a Heʻeia fish pond. So what are their environmental characteristics that need to be looked at for diatom abundance? So I've also shown that diatom abundance differs spatially across the local i'a, and even more pronounced when we look at the differences between seasons spatially. The pattern changes a lot. So in the future, what I'm going to do is explore more techniques to look at multiple parameters and their effects on diatom abundance, or how they're trending together. And I also want to compare other methods to this method, this qPCR method, for measuring diatom abundance. Also in the future, there's going to be survey of gut contents using similar techniques. That's going to be very exciting. So mahalo. I'd like to mahalo all you guys for listening. Mahalo ke akua, mahalo mai ohana, mahalo all these people up here that have helped me both academically, professionally, and personally. Aligato Lab, Paipaio Heʻeia, Nelson Lab, Heʻeia National Estuary and Research Reserve, and Seymour Hale, and anybody else that's in this web connections, mahalo. Okay, and I guess a ui after. So, mahalo.
Thanks, Aka.
Yeah.
That was awesome. I think I'm supposed to introduce our next speaker, Sarah Tucker. She is a PhD student at HIMB in the Rapé Lab. She's also a Margaret A. Davidson Fellow through NOAA with us at National Estuary Research Reserve. And now I think Sarah's going to take us out into Kaneohe Bay and beyond.
Thanks, Shimi. Let me just share screen. I want to say thank you to all the people on the mainland who are in this audience today. It's quite late right now. I appreciate you guys coming to check out our work. And yeah, thank you guys all for being here. Let's see. Okay, so I'm gonna jump right in by thanking the people that I get to work with. I'm very, very fortunate to be in the Rapé Lab and have this amazing support network. All of these data are created through a team effort. I'd like to thank my mentors, Mike Rapé and Shimi Rii, Kelly Friel, who's worked with me on the time series in the Rapé Lab, has been an extremely great partner in crime. Paipai Ohia, Hile, and Kli'i, you guys have been amazing. Everyone at the NERR, Kavika, Katie, Fred, Rosie Elgato, we're looking at synergies where we can collaborate more, and I really appreciate the work that her lab does. And then the Kapuna and the community who have made the Heʻeia NERR possible. They put in all the effort to get this designation and to continue to restore and take care of this space. And I feel honored to be able to work and learn within this environment. So I'm gonna be talking about phytoplankton tonight, and I'm gonna probably reiterate some of the things that we've already talked about, but just so we keep it very general, phytoplankton are single-celled organisms that use sunlight to create their energy. And so they're microscopic solar powerhouses, and they produce 50% of the oxygen in the air that you breathe. So a term that's becoming more popular is take a breath and thank the ocean. And in this process of conducting photosynthesis, phytoplankton actually support the entire marine food web. So they take up sunlight and nutrients and carbon dioxide, and they're able to turn carbon dioxide from this inorganic form, from this gas, into the cellular body mass that forms the phytoplankton. And so being able to take carbon from this inorganic form and turn it into something organic allows it to be then moved up the food chain. So phytoplankton will create sugars that bacteria can feed on. The cellular body walls of the phytoplankton will be grazed upon by zooplankton. Zooplankton will be fed on by fish, and then smaller fish will be fed on by larger fish, all the way up the food chain. And what this comes down to is, without phytoplankton and all the productivity, our entire food web would crumble. And so we've already been introduced to diatoms, the beautiful, amazing organisms that live in a glass shell. Another type of phytoplankton group is our flagellates, and this is kind of a medley of taxonomic groups, such as diatoms and promesiophytes and ciliates. We also have cyanobacteria, such as Synechococcus and Prochlorococcus. So here I just list a couple of the really abundant groups that we see in our oceans. And I've listed them in decreasing size. So diatoms are actually the largest, and Prochlorococcus are the smallest within our ocean. And this is important because diatoms, as being in a larger size, actually have a higher nutrient requirement. Now, it's important because nutrients in our ocean are very limited. Most of our oceans are really nutrient poor. They're actually sometimes referred to as deserts. However, nutrients can increase due to upwelling, which would be water coming up from depth, from the dark ocean, way, way deep down, very nutrient rich in the dark, to the surface. And when it comes up to the surface, it's in an area with more light and now becomes accessible to phytoplankton. So upwelling is one process where we have an increase of nutrients. And then land processes is another important process that increases nutrients and increases phytoplankton abundance. So you can think of, pollution such as fertilizer pollution, increasing nutrients near land. Also, rivers and streams will carry soils with them, and that'll introduce a lot of nutrients into the marine environment. And traditional Hawaiian resource management systems actually took advantage of this increased nutrients from land to create abundant aquaculture systems. So we've already talked a little bit about auapuas, and those are traditional land divisions that create this key unit of resource management. And that includes all these different ecosystems from the mountains to the ocean. So it includes mountains, wetlands, usually a fish pond and an ocean. And here's a photo of the Heʻeia fish pond in 1928. And for those in the audience who don't know, Heʻeia Fish Pond is actually between 600 and 800 years old. So it's pretty amazing to be able to work in a place that has so much history. So in the mountains, fresh water comes down and is introduced into wetlands where there's often lots of different crops growing, sometimes taro. When the fresh water rolls into the wetlands, it brings with it nutrients and sediment. And a lot of times the sediment gets trapped, but the nutrients move on. They move on into the fish pond. And this is really important because the increased nutrients leads to an increased phytoplankton abundance and also an increase in the size of phytoplankton. So that'll allow for, there'll be enough nutrients to sustain those large diatoms that Aka talked about. Whereas in some of our ocean waters, there'll be much less of those diatoms because there's not enough nutrients there. And this all leads to more fish, right? Which is the goal of maximizing the ecosystem services of the fish pond. But there's also exchange with seawater. And that's because the Heʻeia Fish Pond is a fish pond that it has stone walls, and then it has these gates. So a couple of times we said the word makaha earlier in the presentations, and makaha are these gates where water can flow through. So the fresh water can flow out and the seawater can come in, and it creates this exchange of phytoplankton. And this is really where my interest lies. So I'm really interested in the interconnectedness of the fish pond and the surrounding ocean. And my goal is to characterize phytoplankton abundance and composition within these interconnected environments over time. And I'm using time series sampling, which is where you come back to the same point and you sample it again and again at some sort of time interval. And the time interval that I've been using is monthly. So I've been fortunate enough to be within the Repay Lab, and we started a time series back in 2017. And this time series is sampling for things like phytoplankton, nutrients. The whole list is here on the slide, but the important part is that we're getting out there every month since August of 2017 and sampling at these black circles. So these represents the sites we've been sampling at. And with the fellowship that I just started, the Davidson Fellowship that I started in September, I started sampling within the Heʻeia Fish Pond, which is here in purple. And the two sampling sites that I'm sampling at are those in orange right here. And so by sampling these sites within the fish pond on the same day that we're sampling all the other sites throughout Kaneohe Bay and the offshore, we're able to really capture this gradient, this environmental gradient, and understand how phytoplankton are changing with the different environments. It's also a lot of work. So the data I'm going to talk about now are largely from what I've been working on at the 10 sites within the Kaneohe Bay time series, just because I don't have a robust enough data set for within the fish pond. But I do want to point out here on this map of Kaneohe Bay that this is the fish pond right here. This is the Hawaii Institute of Marine Biology, where my lab, the Repay Lab, is situated. And what I'm showing here are the 10 sampling sites in the circles, and the darker colors mean an increase. So here I'm showing average phytoplankton biomass. So this is how much phytoplankton is present. And what you can see is that as you move into the near shore, there's more phytoplankton, right? I said that would happen. The near land, the nutrients increase, and so the phytoplankton abundance increase. But we want to characterize what phytoplankton are where. And so we also looked at Synechococcus and the abundance of Synechococcus at these 10 sites. And again, it increases as we move near shore. You can see in the coastal environment, there is some variation, which is really interesting and something that we hope to look at a little bit more closely. And then when we go to Prochlorococcus, which I said was the smallest phytoplankton that we have, Prochlorococcus is usually limited to really nutrient poor ocean waters. We see that it's really not that abundant within the bay. It gets slightly more abundant at these two sites, and then it really only becomes abundant as we move the furthest offshore that we were sampling. And so what I'm showing here is that we have two different phytoplankton dominating our samples. So within coastal Kaneohe Bay, we have Synechococcus being the most abundant, and offshore we have Prochlorococcus being the most abundant. I'm also showing that these data have led us to do some analyses, and we did some clustering analyses based off of phytoplankton biomass and cell counts, but also pH and salinity. And we were able to reveal that we have 3 distinct types of environments. So we have what we're calling our coastal environment. We have our transition environment. So it doesn't always look very offshore. Sometimes it's in between the coastal and the offshore. And then we have our offshore environment. And when we look at significant differences among our biogeochemical variables, so the different things that we're measuring when we go out, almost all of them, except for temperature and ammonia, are significantly different across these different environment types. So they're really unique. And I list here what is significant, and I just want to point out that I am doing analyses of community composition, and those are unique across the 3 different environment types, but I'm not showing those data today. The next thing is once we have our different understanding spatially of how to look at our sites, we can then zoom in within one environment. So we can zoom into the coastal environment and ask how are the phytoplankton changing over time? So getting at that seasonality question that Aka is also interested in. And I took a slightly different approach. So I used a model to look at 10 years worth of temperature data within Kaneohe Bay. And then it was able to tell me to break it down into 4 different seasons. And here you can see temperature plotted on the y-axis, and then month. So April is the beginning of our spring season. That's how the model broke it up. And then we have July as the beginning of our summer, October as the beginning of our fall, and December as the beginning of our winter. And I just want to point out that this also aligns really well with dry season and wet season. And that's super important, not only because now I can talk a little bit more cohesively with the data Aka's producing, but also this is, there's Hawaiian lunar calendars that for fishing and farming that follow this dry season, this wet season. And so, so it takes our scientific data and these analyses that I'm doing and puts them into a similar type of language that we can talk about with the observations that are being made by the fish pond. So we've seen quite a few different examples of seasonality within the coastal environment. And this is just one example. At the beginning of the dry season, we see increased salinity, increased Synechococcus and bacteria cell counts, and increased phosphate. But really what we want to be able to do is use these data to best understand and manage the fish pond. And so that's kind of where we're going from here, is we're making these observations out in the bay, and we're also thinking about this with the data we're currently collecting in the fish pond, and we're having these conversations about what they mean and where can they be applied. And this is all extremely important and very timely, because our oceans are actually becoming more nutrient poor. Climate change, in particular, the increase of temperature of the ocean, is creating increased ocean stratification. And that's just jargon to say that there's become the nutrients that are way, way down deep in our ocean can't move up to the sunlit ocean, and they're being trapped at depth. And so this is actually already leading to a 6% annual decline in the productivity of phytoplankton. But it's projected that there'll be a 14% decline in zooplankton by 2100. So, the impact is modeled to be moving up the food chain. As we talked about, these are so interconnected. More productive phytoplankton means a more productive food web. And what we're expecting is that with climate change, we're looking at our food web becoming less productive. So our data are helping to provide a baseline that allows for future comparisons to detect or even to model changes due to this increased ocean stratification, as an effect of climate change. And I want to point out that I think a lot of people are starting to think more holistically about the food chain, and I think this is some really exciting work to point out that these people are basically modeling the Hawaii Big Eye tuna fishery based off of phytoplankton size. So there's a lot of people out there thinking about the same things, and this is really exciting. We're starting to acknowledge that, you know, phytoplankton play a huge role in the atmosphere, in the air that we breathe, in the food that we eat, and in the ecosystems that we want to be healthy. And so with that, I have a bunch of funding to thank, and I just wanted to thank everyone here tonight. And I guess we will move on to any questions.
Okay, thank you, Sarah. So the floor is open now for any questions. If you guys have a question, you guys can turn your mic on, or maybe wave your hand, and then we can address it. So the floor is open for any questions.
Taylor, is that a hand up?
I have a quick question.
Yeah.
So I was just wondering, I guess, how do you guys, how do the graduate students here, how do you work together on these projects? Like, do you guys, how do you coordinate some of your research so that you can kind of overlap or at least discuss things? I'm just curious how you guys work this out, because this is a lot of research and a lot of moving parts.
I think Shimi might be the best to answer that.
Sure. Yeah, thanks for that, Kelly. It is a lot of moving parts. You know, so one of the things that center is the NERR does the monthly water quality monitoring, and you know, several of the grad students participate in that. But I think in general, like specifically for Sarah and Aka, you know, they were their research really they were like very separate, but also complemented each other. And I think just like, you know, getting in the same space and talking about it, you know, and not necessarily changing their research, but also just like finding synergies, like Sarah just said, you know, Sarah said in her talk about how they can help analyze and talk about how, you know, if they have certain samples that can be analyzed for certain things, you know, that hasn't been analyzed, or some analysis or something like that. So yeah, just trying to, and then for my part, you know, trying to make these connections happen and just having the conversations.
Yeah, that's great. Thank you, Shimi.
Yeah, thank you.
I see Taylor has her hand up. So Taylor, you can ask them a question.
Yeah, thanks for the awesome talks, everybody. Those were really interesting. I'm from the mainland, so it's really cool to learn about a different NERR. So yeah, this could probably be for either Aka or Sarah. But I was curious what the residence time is like in the fish pond, like how long the water is in there, or whether it gets flushed out with every tide. And then kind of a second part to that is, do you see more freshwater or marine types of phytoplankton in the fish pond?
Okay, so yeah, the fish pond, residence time, I use a figure of 104 hours. So that's like, what, like about 4.25 days. And that's for like neap tide. And then about half that for the spring tide. And yeah, that's one of the comparisons that I used to make across the time series. And as for the types of plankton, I'm not very sure. Yeah, I haven't looked at composition of the populations. So, I mean, Sarah has, so maybe she knows.
I actually haven't analyzed the DNA samples of the fish pond yet, but I do know that in comparison to the bay, there's just a lot more microbes there. So, you know, in particular, like bacteria. There's a lot more bacteria in the pond, larger phytoplankton in general. Yeah, it'll be curious. I think, so I have 2 sampling sites in the pond, and I have one near the stream, and I have one at one of the makahas, which so it's the gate that has entrance with the ocean. And so it'll be really interesting to compare those two, because I think the answer to your question is where do you sample in the pond? Because if you sample near, I believe if you sample near the entrance from the stream, then you're going to get a lot more freshwater type phytoplankton. And if you sample near the ocean, you'll get more. And I think that's kind of evidence of what Aka was seeing with the salinity as well. That's a really good question, though.
Awesome, thank you.
So I got a question from Annie Rosa. She asks, for any of the speakers, how does your interface with the community shape the research that you do? So I guess any of you can answer that. Sarah or Aka.
Oh. Yeah, so for the relationship with the community, how it affects my research is I just like to talk story with them a lot. And just through that, you kind of you know, get hints of what's important to them. You know, you just, I guess you become encultured, right? You kind of like your perspective changes the more you interact with them. So that's how it has affected like the questions that I ask and stuff like that. Yeah. But yeah, they're definitely an important part. Definitely.
Yeah, I would say for me, I get a lot of questions that I've never thought about, and I think it makes me more aware of my research. Yeah, and I think it's really helpful to actually communicate with the community to know more about what their interest is. I'd like to do more of that moving forward. A little bit difficult right now during COVID, but definitely something I hope to do more. And work days really help to provide an opportunity for that.
And I just want to add that you know, they're the ones who know everything, right? They're there every day and know more. So I think, you know, like Aka said, just talking story with them and just being there and observing helps us to really shape our questions that help them to translate our results into actual management decisions.
So it's about 7:33. So I think I'm just gonna wrap it up. But if you guys have any other questions, feel free to send it to our Hanauma account or contact the Heʻeia NERR program. But with that, I want to thank Shimi, Aka, and Sarah for interesting talks tonight, and hope you guys all can make it for next week's talk for another round of 3 consecutive talks at our next Thursday Hanauma Talk seminar series on, I believe, the 28th. So with that, thank you very much. Mahalo, and everybody have a great night.
Thanks, Gavin.
Yep. Thank you.